Hello, my topic is hemoglobin. Hemoglobin is a red coloring matter of blood which is present in the red blood cells. It is a conjugated protein consisting of a heme and globin. It has a molecular weight of 64,450 daltons and can combine with oxygen and act in transport mechanism of oxygen within the blood. It contains 4 gram atoms of iron per mole in ferrous state. The structure of hemoglobin can be classified under two types. The first one is the structure of heme, that is the prosthetic group, and the second one is the structure of globin, that is the protein part apoprotein. Coming to the structure of heme, it is an iron porphyrin. Porphyrins are cyclic compounds that contain 4 pyrrole rings, hence it's called as tetrapyrrole structure. These rings are named 1 to 4 and are linked through methylene bridges or methyldiene bridges. The outer carbon atoms which are not linked through methyldiene bridges are numbered from 1 to 8, and the methyldiene bridges are designated as alpha, beta, gamma, delta respectively. The structure of heme consists of iron at the center in its ferrous state and is bound to the nitrogen atom of the pyrrole rings. Iron is also linked internally to the nitrogen atom of imidazole ring of histidine of polypeptide chains. The third and fourth pyrrole rings consist of propionic acid in their sixth and seventh position, which are linked to the amino acids arginine and lysine of the polypeptide chain respectively. Next, coming to the structure of globin. The globin of hemoglobin is a protein which is composed of 4 parallel layers of closely packed polypeptide chains. Two of the chains have identical amino acid composition of 141 amino acids. The other two chains may be of 4 polypeptide chains designated as beta, gamma, delta, and epsilon, each of one having 146 amino acids. The total number of amino acids in globin is 574. Alpha chains have valine, leucine, serine in N-terminal residues and lysine, tyrosine, arginine in C-terminal residues. Beta chains have valine, histidine, leucine in N-terminal residues and lysine, tyrosine, histidine in C-terminal residues. Gamma chains have glycine, histidine, phenylalanine in N-terminal residues and arginine, tyrosine, histidine in C-terminal residues. Hemoglobin molecule and its subunits contain hydrophobic amino acids internally and hydrophilic amino acids on the surfaces. So these form heme pockets. In heme pockets, alpha subunits are of size which let oxygen molecule, but the oxygen molecule entry is prohibited in beta subunit, which is blocked by valine residue. Coming to the properties of hemoglobin. First one is oxyhemoglobin. Hemoglobin combines with oxygen to form oxyhemoglobin. When the hemoglobin is exposed to air, it takes up 2 atoms of oxygen for each atom of ferrous ion present. Thus, hemoglobin will take up 4 molecules of oxygen. In low oxygen tension, when the amount of oxygen in body is reduced, oxyhemoglobin gives up oxygen which is carried out by blood to different parts of body. Next is formation of carbamino compounds. Hemoglobin reacts with carbon dioxide to form carbamino compounds. The third one is the reaction with carbon monoxide. Hemoglobin reacts with carbon monoxide to form carboxyhemoglobin. Carboxyhemoglobin is stable and prevents the formation of oxyhemoglobin. So inhalation of even small amounts of carbon dioxide is highly dangerous. The next is buffering action. One molecule of hemoglobin contains 35 histidine residues. Histidine exerts its buffering action through its basic imidazole ring. Hence, hemoglobin plays an important role in regulating the acid-base balance in blood. Formation of methemoglobin. Methemoglobin is formed by oxidation of hemoglobin by mild oxidizing agents like potassium ferricyanide. The ferrous ion in the hemoglobin molecule is oxidized to ferric ion. Methemoglobin cannot carry oxygen in blood. It is formed by action of some drugs. This condition is found in individuals having inborn errors of metabolism. This can be reduced to hemoglobin by vitamin C, which is used in the treatment of methemoglobinemia. Cyanomethemoglobin. It is formed by the addition of cyanide to methemoglobin. It has bright red color. Sulfhemoglobin. It is formed by administration of certain drugs. It continues to remain in blood and cannot be reconverted into hemoglobin. Next is the biosynthesis of hemoglobin. The biosynthesis of hemoglobin takes place in bone marrow in erythroid cell during its development to erythrocyte. It starts appearing at stage 2, that is early normoblast, and the synthesis is complete when the stage reaches 4, that is the late normoblast. Iron and ferrous state is incorporated into photoporphyrin to form heme. The heme gets attached to the newly synthesized globin to form hemoglobin. The iron of heme is coordinated to 2-imidazole nitrogen of histidine at position 38 and 87 in alpha chains and 63 and 92 in beta chains. Transportation provided by hemoglobin. Hemoglobin transports carbon dioxide and protons to lungs after releasing oxygen to the tissues. Hemoglobin can bind carbon dioxide directly when oxygen is released, and carbon dioxide reacts with the amino terminal, that is A amino groups of hemoglobin, forming a carbamate and releasing protons. The amino terminal is converted from a positive to negative charge, favoring salt bridge formation between the A and P chains. At lungs, hemoglobin is oxygenated, being accompanied by expulsion and subsequent expiration of carbon dioxide. Carbon dioxide is absorbed in blood, and the carbonic anhydride in erythrocytes catalyzes the formation of carbonic acid, which is rapidly dissociated into bicarbonate and a proton. A buffering system absorbs these excess protons to avoid the increasing acidity of blood. Hemoglobin binds 2 protons for every 4 oxygen molecules. In the lungs, the process is reversed, that is, when oxygen binds to deoxygenated hemoglobin, protons are released and combines with bicarbonate, forming carbonic acid, which is exalted. Thus, the binding of oxygen forces the exaltation of carbon dioxide. This reversible phenomena is called as the Bohr effect. Varieties of human hemoglobin. Normal adult hemoglobin or hemoglobin A has a molecular weight of 64,456 daltons and contains two pairs of peptide chains alpha and beta, of which alpha chain contains 141 amino acids and beta chain contains 146 amino acids. Fetal hemoglobin F is present in very small quantity. All the normal human hemoglobins possess a common half molecule, that is a pair of peptide chains. The other half consists of a pair of different types of peptide chains, one type of each hemoglobin. Hemoglobin A2 has two delta chains and hemoglobin F has two gamma chains. Both types of chains contain 136 amino acids and thus are of same length as beta chain. Fetal hemoglobin. Fetal hemoglobin comprises 50 to 90% of total hemoglobin in newborn. It takes up oxygen more readily at low oxygen tensions and releases carbon dioxide more readily than adult hemoglobin. It is more resistant to denaturation by alkali and is more susceptible to conversion of methemoglobin by nitrates. Hemoglobin F is gradually replaced by hemoglobin A during the first 6 months of extrauterine life. High concentration of hemoglobin F after 2 years of age occurs in various types of anemia, example sickle cell anemia and thalassemia. Abnormal hemoglobin. Over 100 different types of abnormal hemoglobins have been described. Some of these are easily differentiated by their electrophoretic mobilities and have given rise to the concept of molecular disease, which explains that a defective gene may direct the formation of a molecule similar to normal molecule but different from shape, composition, and electric charge. One amino acid of normal hemoglobin is replaced by other amino acid, that is, acidic amino acid is replaced by basic or neutral amino acid for the formation of abnormal hemoglobin. So this was some brief information about the molecule hemoglobin. Thank you.